not. We're just going to learn the major ones. There's so many that I don't know if you could handle all of them.
It would make your head itch.
It's a lot of muscles.
Things are going to start happening to me now.
You know what's funny? Just in the last couple of years, they just discovered 2 new muscles. How about that?
What do they do?
They're in your forehead, and they kind of help move your skin of your forehead. And they're real tiny, and nobody had ever seen them before.
Awesome.
Somebody was doing a cadaver dissection, and
Like, what's
somehow
that?
saw them, and were like, what are these? And they found out it's a muscle that no one had ever seen.
Things are going to start happening to me now.
What movie? The Jerk. You ever seen The Jerk?
Mm-hmm.
No.
Excellent.
No.
Very fun.
Oh, wow.
Okay, so we're going to learn about the structure of a muscle and how it works today. It's pretty it's pretty difficult to understand. There's a lot to it, but I'll give you the major stuff here. Here's a muscle. You can see a muscle is attached to a bone by what we call a tendon. Can you tell what bone that is just by looking?
Femur.
Ha, y'all have learned something.
Femur.
Yes, that's the femur, and you see, the reason why the femur has all these bumps and shapes and protrusions on it is for muscle attachments. And so the muscle, the calf muscle here is attached to the femur on one end there, and it's attached by a tendon. So if you tear a tendon, you tear the attachment between a muscle and a bone, and then that muscle can't move the bone anymore. That's a problem. Usually people will get partial tears and not full tears. And then it hurts every time you use the muscle because it pulls at the tear. And there's nerve fibers running through there.
Could it literally?
It could completely tear off, or it could heal up. So you have to treat those, go to the doctor and see what they say to do.
What other bone does that connect to?
On the other end, it connects to the heel of the calcaneus through the Achilles tendon.
So that's a long muscle.
That's right.
So
The calf is a long muscle. So anyway, if you look at the structure of the muscle here, it is divided. It has a covering around it called the fascia. And if you cut into the muscle, you see it's divided into groups called fascicles. That's a fascicle, that's a fascicle, that's a fascicle. It's a big bunch of muscle cells. And they've pulled out one of the fascicles so you can see it up close. And it's like it's made of a bunch of tiny cables. You know, if you cut a big phone cable and look inside it, there's a bunch of little tiny cables inside it. You ever done that? Probably not, but you can imagine. And so each little circle here is a muscle cell. They pulled out a single muscle cell. Now muscle cells are real long and thin. They're like strings. And so they don't really call them muscle cells, they call them muscle fibers because they're so long. And so this is a single muscle fiber here. It helps make up a fascicle. A fascicle is made up of many muscle fibers, and a muscle is made up of many fascicles. And the muscle fiber itself is a long cell that has many nuclei. There's a nucleus. You know, the nucleus contains DNA. You should know that from regular biology. And there are multiple nuclei just spread down the muscle fiber. And there's an outer membrane to the muscle cell. It's called the sarcolemma. And by the way, some of these words are going to be on your ID sheet that's coming up. You'll get an ID sheet pretty soon. We're not going to work on it today, but we'll start on next week. So the sarcolemma is the membrane around the muscle cell. There's a nucleus, and there are multiple nuclei spread all down the muscle cell. This area right here is called the Sarcoplasmic Reticulum. You probably learned in regular biology about Endoplasmic Reticulum. Do you remember that? Inside a cell? Endoplasmic reticulum. Do you remember, anyone remember from regular biology what endoplasmic reticulum does inside a cell?
Ribosomes.
What's that?
It makes ribosomes?
It has, sometimes it has ribosomes on it. Rough endoplasmic
Does it make
reticulum.
protein?
The ribosomes make proteins. What do you think the ribosomes do with the proteins after they make it? Package them in what? What do you think? And where do the vesicles originate? The endoplasmic reticulum. Yes! I had to fish for that, but I finally got it. A fishing reference for you. So, in a cell, the endoplasmic reticulum is a storage area. In the muscle cell, the sarcoplasmic reticulum is a storage area too. It's a storage area for calcium. Most of the calcium that you eat, or a lot of it, goes into the sarcoplasmic reticulum. If you have leftover calcium that you don't need right away in the sarcoplasmic reticulum, it's stored away in your bones. But calcium is very important, you'll see, for how the muscle works. Now, inside that, you have all these fibers. They're called myofibrils. And they're made up of two types of protein. The thin filaments here are called actin, and they're colored blue in this picture. And the thick filaments here are called myosin, and they're colored purple. And actin and myosin are going to work together to help a muscle move. Okay? Some of y'all don't seem too interested in this. Well, you wouldn't be able to move without it. Yes?
Is the actin and myosin, is it like, one pulls this one pulls the other to determine which way the muscle goes?
No, but I'll show you how they work. They do pull.
Which one, like,
The myosin pulls on the actin, and it helps make the muscle smaller. That's what we're going to do in about 10 seconds here. So I'll show you how that works. Kind of complicated. So these thin and thick filaments, here you see it up close, they're protein. Muscle, have you ever heard muscle's got a lot of protein in it? If you eat meat, you're eating the muscle of a cow or a pig or whatever, deer or whatever kind of meat you're eating. Human, I don't know what y'all eat. But anyway, what's that? No, me neither. But, you go to a restaurant, you don't know what they're serving. Anyway, you can see the dark myosin here and the blue actin here. The purple is myosin and the blue is actin. And you can see how they overlap with one another. They overlap like my fingers are kind of overlapped here. And the way they overlap gives them the ability to shorten in length or contract. And you see when I do that, my thumbs get closer to one another. See? That's what muscles can do. And when you kind of look at this from afar, what you'll see dark where the myosin is, and then a lighter color where it's only actin. So you see dark, light, light. And this has kind of been named by scientists. Before they knew what this was, they saw the dark area they called the A band, and the light area they called the I band. And you can see this little zigzag line here they called the Z line. They weren't sure what this stuff was. They knew they could see lines on the muscle. And if you look at it from afar, like we did under our microscopes, you see those lines. Do y'all remember seeing lines when you looked at muscle? Mm-hmm. Striations? Do y'all remember that? That was this dark area, light area, dark area, light area, and it moves all through the muscle cell. And that's what you were looking at. This is what it looks like. Dark, light, dark, light. And so how does the muscle work? That's what we're going to get into now. Here's a muscle cell, another view of it. And here's the sarcolemma, the covering. And there are little holes in it. Openings into transverse tubules, these little holes here. And what they do is they allow an electrical signal that comes from a nerve to penetrate down deep into the muscle. And the electrical impulse is going to stimulate the sarcoplasmic reticulum, which is brown here, to release its calcium. The calcium leaks down into the actin and myosin fibers and causes the myosin to start to pull on the actin to shorten the muscle. And the best way I can show that to you, how it works, is with video. Here it shows a nerve coming up here and attaching to the muscle.
So, like, if we were to want to move here right now, that would actually create enough calcium that quick for us to move our muscle.
That's correct. Yeah, you can say, I can say, I want to move my arm, and you start a nerve signal in your brain, and it shoots a signal down to your muscle, and the muscle contracts and pulls your arm up, and you can do it like that because it's so fast. So fast. That's how I dance at Ziggy's. They're all like, what? They're like, man, it's good. So let me show you the video here. It's hard to understand any of this without the video. When I was a kid, we didn't have video. We had to learn this without any of the videos. It was impossible. We didn't learn anything. Oh, I need sound.
Neurotransmitters are...
Okay, so here we go. This is a muscle fiber. Are you with me, McLean?
I'm with y'all.
This is a nerve cell coming up.
Release. They stimulate the propagation of an action to...
You know what? Let's not listen to him. He's very monotone. Let me do the talking here. If I can play it. Okay, so here comes the signal. That's the red thing coming up. And the signal moves down these T tubules. You see that red thing going down into the muscle. And it penetrates deep into the muscle. All of these fibers are stimulated by this one nerve. And here we see the red nerve signal coming down. And let me pause it right here. These yellow things you see are calcium ions that are stored in this blue area. That's the sarcoplasmic reticulum, the calcium storage that's in your muscle. And so when the signal comes in, the signal stimulates the blue areas, sarcoplasmic reticulum, to release calcium. And that's what it's showing here. See the calcium being released? The calcium sinks down into the muscle fiber. Now, we look up close at the muscle fiber. This is actin. It's the thin protein filament. This down here is myosin. It's the thick protein filament. This is called a myosin cross bridge. It's like an oar that, you know, have you ever rowed a boat before? You got oars, right? And the oars can pull something. That's exactly what these things do. This is part of the myosin, and it's going to reach up and grab the actin and pull on it. And I'm going to show you how that works here. But first, the first thing we got to do is the calcium is going to sink down here, and the calcium ion is going to bind to this green protein here that is on the actin. The green protein is called troponin. I don't know why they call it that, but they do. Troponin. And if you notice, the green protein here, troponin, is attached to this green rope-like protein that's called tropomyosin. They show this structure, this setup on page 182. The troponin and tropomyosin, it looks a little bit different. But what's going to happen is the calcium is going to bind to the troponin, and then it's going to cause this rope-like structure to move. So let's watch that happen. Calcium binds to the troponin, and there's the tropomyosin, the rope-like structure, and that's going to move. Go ahead, move. Yep, see it moved?
Mm-hmm.
Did y'all see that? That was very exciting. Now, here, now you see, after that rope-like molecule moves, it exposes these things that look like birds' beaks, called myosin binding sites. And those are for this cross bridge to reach up and grab. So now this cross bridge right here, this myosin cross bridge is going to reach up and grab. See, it grabs on, and it's going to pull with the help of an ATP molecule. If you remember your biology, ATP is an energy molecule. It just came off, and now we get a pull. And now more ATP is going to come, and it's going to cause it to release and go up and pull again. And this is happening in multiple places so that you're pulling along that actin.
Looks like an assembly.
Kind of, yeah. You got many things working together. Let me back this up a little bit. Let's watch the whole thing again. Here comes a nerve signal. The signal goes down the T tubules. There's a lot of multiple choice questions about this on the next test. Signal goes down these T tubules, causing calcium to be released from the Sarcoplasmic Reticulum. There's the calcium being released. The calcium then binds to the troponin, bam, causing the tropomyosin to move, exposing the myosin binding sites so that the myosin cross bridge can reach up and pull using ATP as an energy source. Many myosin cross bridges working together can cause the muscle to contract as a whole. Because this is happening in multiple places along the muscle fiber. Are y'all with me here?
But I mean, it's going like 1,000 times faster than
It's going 1,000 times faster than that.
maybe even.
And the muscle shortens. Adenosine triphosphate. So what happens when you stop sending the message to the muscle? Well, when you stop sending the message to the muscle, the calcium goes away. And the myosin binding sites get covered up. Now the cross bridge can't attach. It's trying to attach, but it can't. Oh. And the whole thing slides back. And your muscle relaxes. And they just run through the whole thing again here. Kind of a review here. So, all of this happening in many places on a muscle. Now, imagine one muscle has about 1 billion of these little cross bridges on it. So there's a lot of activity going on when you contract a muscle. And this kind of shows it, shows the whole thing at work here.
Short fiber.
Look at the stripes. You see the
On
stripes?
more detailed examination, however, this contraction is seen to be the result of the much finer shortening of the striations themselves. The substructures responsible for this movement could not be adequately observed until the development of the electron microscope. Then the complex internal arrangement of the muscle fiber was revealed. It was found that the fiber is made up of small substructures, fibrils. And the fibrils, in turn, are composed of still smaller units, which were named filaments. Two kinds of filaments could be distinguished. Relatively thin ones, which form the light stripes, and relatively thick ones, which form the dark stripes. This accounts for the striated appearance of the fibril. Examination of a fibril in cross-section shows that the component filaments form a definite pattern. This pattern is due to the regular arrangement of the thick and thin filaments. The thick filaments on chemical analysis were found to be composed of the protein myosin, while the thin filaments are composed of the protein actin. Based on the visual evidence of electron microscope studies, a working model of how the filaments behave during contraction was constructed. It suggests that during contraction, the filaments slide over and past one another. This is the sliding filament theory of muscular contraction.
Hold on, one more video and it'll be perfectly understandable.
One nerve impulse causes many muscle cells to contract, while another triggers just one or two. In fact, the links between our nerves and our muscles are like a kind of fixed wiring. A nerve and the muscle fibers it controls are together called a motor unit. In the fingers, a motor unit may be as small as 10 fibers to one nerve. But in the bigger muscles of the body, which need strength rather than delicacy, a motor unit may be one nerve controlling hundreds of muscle fibers. But what happens inside an individual muscle when it receives a nerve message telling it to contract? To find out, we need to penetrate down to the level of the individual molecules in a muscle cell. Here's a single muscle fiber, and the secret of how it shortens is hidden in these stripes that run across it. Each of them marks a tiny zone of contraction. The fiber is making a series of twitches in response to a nerve signal. Each line marks the place where millions of stick-like molecules are lined up side by side. These molecules interweave with others lined up facing them. The fiber shortens when the individual pink molecules pull against blue arms that interweave among them. When the blue molecules are as close as possible to each other, the muscle is fully contracted.
So this is showing it. When I was showing it on the other video, it was horizontal. This is showing it vertical. But that's the same thing. The pink is the myosin, and the blue is the actin, and the myosin is pulling the actin so it goes closer together. I think that shows it best because you understand that it's a whole bunch of these units working together.
Like molecules are lined up side by side. These molecules interweave with others lined up facing them. The fiber shortens when the individual pink molecules pull against blue arms that interweave among them. When the blue molecules are as close as possible to each other, the muscle is fully contracted. This can produce an overall shortening of 50% or so in a muscle, like the biceps.
So when you make a muscle, you're just sending a message to tell the muscle to contract. And everything shortens up together, and that's why it's bigger and thicker. So if you want to straighten that arm back out, you just contract the other muscle on the other side, the triceps, and that pulls it back. Contract this, pulls it up, contract this, pulls it back. Every motion has actually two muscles, one that goes in one direction and one that goes in the other. Using some ATP, y'all. I'm tired. Yeah.
I was reading right here, it says creatine. Is that why like a lot of athletes they used to drink that?
Yeah, creatine is a chemical that's in the muscles themselves, and actually creatine can restore the ATP really fast. It's kind of like creatine's kind of like a battery. So it takes a long time to restore ATP, to put ATP back together the normal way through the mitochondrion. Creatine phosphate can do that pretty quickly. So it's
you work out, you know, your muscles stretched out and it repairs the signal.
It's not like it's repairing your muscle. It's like it's recharging that ATP that comes off and breaks up. It's the creatine yields some phosphorus to it to put it back together. So creatine is a quick recharger that's located within the muscles. So if you take extra creatine, it's supposed to, be able to charge your muscles back faster. I don't know how well that works, but that's the idea behind it. Okay, so you're supposed to read 8.1 through 8.3 tonight, and it basically explains everything I just talked about. Okay? How good are y'all? Happy Valentine's. That's my Valentine's Day gift to y'all. A reading assignment. Huh? Because I care.